breath.
Yeah, I'm in class now, Padma. Last class, we discussed unequal crossovers. That is between exactly similar looking, similar DNA containing domains once they are duplicated and next to each other. Just a minute. So hope everybody is here. Okay. So between the duplicated domains, crossovers can happen unequally. That means here, if we think this is the left domain and this is the right domain, the synapses in the meiosis should happen left to left and right to right. Mistakes can happen because of misalignment. This misalignment can happen because the DNA content in those domains is exactly the same, complementary. So instead of left aligning with left, left can align with right. So this kind of unequal crossover leads deletion or gain of the domains. Here a copy is deleted, but here one gained happened. Okay, so this is what I mentioned, unequal crossover, by which the chromosomes undergo deletions or duplications. Can you hear me? Can you all hear me? If any concerns, please type it on. Please type it on chat so that I will know what is the problem. Or even you can speak up. So we discussed about red pigment and green pigment, how multiple green pigments can form in one chromatid. And then some of them will have deletions that it do not have a green. So the mammals, especially females, will have two X chromosomes, at least one X chromosome will have compensate the alleles. But whereas if it is one X chromosome, like in males, if it has deletion, that particular male will be colorblind. So we discussed this, how even between the repeats, misalignment can happen. Repeats are exactly, having repetitions of nucleotides, and then they will undergo synapses. Either gaining the copy number in between or deletion of the genes will happen. So most importantly, we discussed inversions, but we did not discuss how the inversions form. Do we have that picture here? It is not there. So in one another class, we can discuss how this kind of unequal crossing overs also will lead to the inversions. And once it is inverted, one chromosome will be normally looped, normally bent like this, and the chromosome that has inversion will loops around so that the numbers would match, so that the loci will match that are inverted and not inverted. So this can be paracentric or pericentric. If there is paracentric, that is centromere is next to the loop, then there will be more severe problems like dicentric chromosomes can form. Dicentric in the sense having two different centromeres. Acentric means there is no centromere. So this kind of chromosomes with the dicentric will undergo breaks because the spindle fibers can exert some kind of pull on them. This spindle fiber is pulling to this pole, and this set of spindle fibers are pulling to this pole. The chromosomes get broken. If it is acentric, that do not have any centromere, so this will get lost. And then if there is centromere within this loop, this can stabilize the crossover. If the crossover is not happening, that is well and good, very good. They can just synapse and separate. The gametes will be normal, maintaining one inverted chromosome, one normal chromosome. If the inversion loop undergoes crossover, these are the problems. One is paracentric and pericentric. Pericentric is crossover is happening, and then there is a centromere there. So that can stabilize quite a bit, but there will be still duplications and deletions. See, instead of A here, there is D here. But here, this D is triplicated in 3 different places. So in another chromosome, it is A and D again, and then D is replaced with S. So A is triplicated. So this kind of pericentric inversions can cause deletions or duplications of genes, not domains. Okay. So the translocations, we discussed reciprocal translocations, the balanced translocations. These are the normal chromosomes. Blue is a kind of chromosomes, red are kind of chromosomes, but blue is migrating to the red chromosome, and then this is heterozygous. Okay, so on the blue is migrating to the red and red is migrating the blue, but one set is normal. Since one set is normal, then we call them heterozygous. Then in the homozygous translocation, that means both chromosomes undergo this. That is very rare. Both chromosomes having a translocation, like both mother and father should be translocation chromosomes. Very rarely it can happen, probably, especially in humans. Whereas generally you can find this kind of translocations where one chromosome is normal, whereas the homologous chromosome have undergone this kind of translocation. So when the synapsis happens, this can form a ring. That ring is called quadrivalent. So the quadrivalent look like this. One is the normal chromosome. This is the normal chromosome. This is the chromosome with this fag end. You have the blue segment on the red chromosome. So it bent in such a way that the blue aligns with the blue and red aligns with the red. So these are called quadrivalents. Why it is quadrivalent? Normally, if it is one single homologous chromosome, it is bivalent. When the 4 chromosomes join together like this because of translocation, this is called quadrivalent. So this quadrivalence can separate anyway during the meiosis. Centromere, the spindle fibers will bind to the centromere and pull to one direction, like this 3rd chromosome and 4th chromosome go to one chromosome, one pole, and then this chromosome and this chromosome, 1 and 2 go to the other pole. Okay, so with this, now all these chromatids that are occurring will have at least one translocation. You see, if these two are going to one pole and becoming 2 different cells, so one blue chromosome and one translocation chromosome will be there in each cell. So all of them will have translocations. Whereas the pull can happen the adjacent two. This is adjacent one. That means the pull is happening sideways. This is adjacent two is these 2 chromosomes will go to one pole, these 2 chromosomes go to one pole, and then the cells will form. One will have normal chromosome, one will have translocation chromosome, and then all the 4 gametes that are resulting in the meiosis will have translocation. Okay, these parents, all of them will have translocation. So a parent having translocation, that means what? In meiosis, when synapsis occurs, they will form quadrivalent. Otherwise, there is no quadrivalent. Okay, here, though there is quadrivalency is there, now the way these spindle fibers are pulling, so the 2 and 3 are diagonally pulled to one pole, and then they are normal chromosomes. 3 is normal chromosome, 2 is normal chromosome. So you can find 50% of the gametes normal, whereas 50% of the gametes have undergone this kind of translocation. So even the translocation parents, one parent is translocation, quadrivalent is forming, but the way they get separated can still form normal gametes. So that is the adjacent, and we discussed about Robertsonian translocation, where chromosome 14 and chromosome 21. Here is chromosome 14, other one chromosome is small. And why this chromosome is long? Because 21 joined here. 21 can piggyback here. You see, this 21 is small chromosome and telocentric, that is, centromere is on top. So this can easily get fused onto the chromosome 14, which is long but telocentric. So two telocentric chromosomes always can attach. So this is one way of happening Down syndrome. Okay. Very accidental touching of two chromosomes. So we discussed last class polyploid. What is polyploid? Having multiple sets of chromosomes. It can happen in asexual way and sexual way. And we discussed in sexual way what happens. The gametes will not undergo meiosis. Okay. The gametes will produce, as it is, gametes with somatic number of chromosomes. Like we humans, we reduce our gametes to 23 chromosomes, but our somatic cells are 46 chromosomes. If meiosis is not happening by mistake, then we will produce gametes with 46 chromosomes, which is fatal in humans, but which is acceptable in plants. That can lead speciation. Okay, that becomes a different species. So in asexual polyploidy, what happens? The sex happen normally. Okay, so the somatic cells undergo meiosis and produce gametes that are having reduced number of chromosomes or half of the chromosomes. But the zygote, which is 2n zygote, can become 4n through a process called endoreduplication or duplication. Some problem in the spindle fiber formation, and then 2n becomes 4n. So either way, either sexual fashion, which is unreduced gametes, or here is reduced gametes, but the zygote became doubled. So both ways it can produce polyploidy. This is all discussed, so we are revising. So I'm talking faster. And then there are autopolyploids, where all are same species, like in mumps, chrysanthemums. N equal to 9, diploid is 2n, which is 18. Tetraploid is 36. Hexaploid is 9 times 6. Octaploid is 9 times 8, that is 72. Decaploid is 9 times 10, that is 90 chromosomes. But same species is getting doubled, tetraploid, that is quadrupled, or hexaploid, or octupled, or decapled. So multiply of several chromosomes. So the plants can withstand. So folks, you have seen, having the larger number of genes, what will happen? There will be more gene expression. That means they will produce more proteins. If they produce more proteins, they are going to have more expression, gene expression. So they will have enhanced trait. So the flower size in mums, in diploids it is very small. In tetraploids it is larger. Hexaploid it is more larger. Octaploid will have really bigger mums. And the biggest mums you can see in the decaploid. Okay. So, but these are all auto, self-polyploids. Same species is getting doubled, quadrupled, 6 times, or octupled, that means 8 times, or decapled, which is 10 times. Whereas allopolyploids is two different species can undergo sex. Two different species, how it can undergo sex? Like humans cannot have sex with chimpanzees or gorillas. We cannot because we are diverged quite a bit. But in plants, this can happen. In plants, what happens? Here is the species A, species B. So accidentally, sex can happen. Both gametes of this gametes of this should not undergo sexual reproduction, but it can happen. And then there is zygote, which is having these chromosomes and this chromosome, one of this chromosome and one of this chromosome. So say this dark blue, this red color chromosome, and then light blue and light red chromosome coming from different species can form a zygote. But that zygote is sterile. That zygote will die. But what happens sometime? That chromosome number in that sterile zygote gets doubled by some mechanism. Okay? Something like endo-reduplication. That is, spindle fibers will not form. You know, even thousands of zygotes are forming like that. In one or two zygotes, a miracle can happen where the chromosomes can get doubled. So now the blue are two, light blue are two, red are two, light red are two. So it becomes totally different species. This is called new allotetraploid species. Allo means what? Different. It is tetraploid, but two different species. So species A and species B coming together through sex. That is, sometimes they produce, say, same number of gametes as it is. Sexual or asexual means. Sexual means the gametes are not undergoing meiosis. Chromosome number is not reduced. And then sex happens between them, and directly this kind of zygote is produced. Or alternatively, those two undergo meiosis normally. You have one blue and one red in the gamete. Here also in other gametes, say in the egg, one blue and one light blue and one light red. But once the zygote is formed, the chromosome number is getting doubled. That is asexual polyploidization. Sexual means the gametes resemble like parental cells, somatic cells. There is no reduction here. So two blue, two red, two light blue, two light red. All of them are forming gametes. And as a result of this, accidental sex can happen between the plants. This accidental sex can lead to new species. So this is how the plants produce different species. Whereas animals, how they have to produce different species? Because in animals, polyploidy is not tolerated. We cannot double our chromosomes or we cannot have sex with a totally different species. Even the closest mammal, like chimpanzee, we cannot have and produce a baby. It is not possible. Sexually, that is nothing, completely incompatible. But the plants can undergo this kind of mysterious sex to produce polyploids. And then polyploids can survive and will have advantage and eventually become different species. So wheat is such species. In wheat, there are many different species actually. The bread wheat, the bread today we eat, is a allopolyploid that has three different species. First species A, species B, fused and produced, and then this again fused with another species. So three different species combined, pyramided, to produce wheat. Whereas cotton has two different species. One cotton is old world cotton, which is Africa and Asia. That cotton is a diploid cotton, normal cotton. You know, that one swept through the oceans when the platonic merger is happening. That time it might have happened, or the seeds flown through the ocean and reached to the New World, to the Mexico. So in Yucatan region of the Mexico, the sex occurred between the cotton A genome and cotton D genome. And then cotton A and cotton D produced a F1, which is AD. AD is not fertile, so it became doubled to become AA and DD. Okay, so that kind of very intriguing things can happen. Like, let me draw it how it looks like. Say if one cotton is AA cotton. This AA cotton is coming from Africa, swimming through the ocean. The seeds flown and entered into Mexico. And then AA cotton and DD cotton, not BB cotton. Okay, DD. Okay, so this produces a normal gamete having A in it. The gamete will have one set of chromosomes, that is, we call it as A genome. And then this will have, gamete will have reduced number of gametes, DD becomes D. And then the normally sex should not happen, but it happens, becomes AD. But this AD is sterile, because A set of chromosomes, D set of chromosomes cannot produce fertile baby, but the doubling can happen. A becomes AA, D becomes DD. Now this is called tetraploid, allotetraploid. So cotton evolved today like that. Had cotton did not undergo this kind of sex, you would not have seen your t-shirts, your cotton socks, cotton t-shirts, or cotton pillows, or even your dollar bills. 90% of your dollar bill is cotton. Okay, so because plants underwent this kind of illegitimate sex, today we have cotton. And today we have bread, wheat bread, because three different wheat species have sex similar way to produce a hexaploid, hexaallopolyploid. So three different species that have undergone sex accidentally. Not very old actually, maybe 10,000 years, 15,000 years. That's it. In the Neolithic, just before Neolithic times, this sex might have happened in the fertile crescent, which is Syria. Okay, so the cotton evolved in Mexico, and wheat evolved in the old world, in Syria or Iran, where this kind of illegitimate sex occurred between different plant species to produce very useful and economically important species. Okay. So the polyploidy is very well conserved in grasses. So what are the grasses? All the cereals that we use. What are the cereals? Rice, wheat, maize, oat, millets are cereals. Sugarcane is a sugar crop. It's not a cereal, but it is still the grass. All these grasses belong to, had one common ancestor some time back. So these grasses are different species. Rice is different from wheat. Everybody knows that. I don't need to teach that. And wheat is different from maize. And then we know sugarcane is different. Candy, sugar is different. Sorghum is different. And oat, rye, there are many millets like that. So all these grains have undergone illegitimate sex to differentiate themselves. So that once we do sequencing, we can understand based on that how this might have happened. So the rice is the smallest genome, and then we can see how the rice genome sequence is shared between different chromosomes of various grasses. So here, for example, rice, is the total genome sequence of rice here. R1A in the sense chromosome 1A, half of it. Chromosome 1B in the sense chromosome 1, half of it. Chromosome 3, three different parts. This is chromosome 2 as just one single unit because it is a small chromosome. So you can see here all rice chromosome segments. And wheat is nothing but wheat chromosome 1 is chromosome 5 of rice, chromosome 10 of rice, and chromosome 5b of rice combined to become wheat chromosome. Similarly, each chromosome, you can relate it to the rice. So that means it is syntonical, collinear, syntony. Syntony means relationship. You can understand the relationship between your chromosomes and chimpanzee chromosome. Okay, how they are different. In fact, very strong. We have exactly same chromosomes almost preserved between chimpanzees and humans. Only at expression level, we have differences. Some of the human genes overexpress, or some of the chimpanzees genes overexpress. So these expression differences cause different genomes, different species. So this is just like a deck of cards, playing cards. You shuffle at a time, you will see the same cards in different orientation. Foxtail millet, sugarcane. Sugarcane is, if you, sugarcane chromosomes are designated like A, B, C, D, E, F, G. E, F, G, yeah. So each chromosome again can be related to rice. You see? So what this indicates, folks, this tells us that all these grass species once upon a time had a common ancestor, and that common ancestor has broke into different species. And then these species evolved by refining various segments of rice in the various chromosomes. And then expression levels differ in such a way. Some genes, rice genes, will express differently here. Rice genes express once they are moved here with the different parts of other chromosomes. They start expressing differently. So these expression differences of genes make them different, totally different cereals. You say, hey, I don't like rice, give me bread. But it is pretty much same. What you are eating, whether you are eating a bowl of rice or a candy or a corn soup or whatever, a millet food or rye or oatmeal, everything, you are eating pretty much same gene product that is shuffled in different fashion. That is really interesting, isn't it? This kind of syntonic groups exist, can be tracked back in grasses. Similarly, in Solanaceae, my lab worked with Solanaceae family, which is important for tomato or peppers or eggplants or petunia, okay? So various other Solanaceae plants, all of them are evolving from the single ancestor. You can relate, same, we have tomatoes and chili peppers have the same genes to make pungency. But if you eat chili peppers, it is hot. You have same genes in tomato also, exactly same genes. But why tomato is not hot? Why it is not pungent? If you want to make salsa, you will take some tomatoes and some peppers and grind them together, right? That is salsa. You can eat with corn chips because it will have nice tangy taste. But you have the same genes in tomato. Why not to make, if you want, you can, using genetic engineering approaches, you can make those tomatoes hotter, pungent. Probably Arushi should do the research like this. Should take all the genes that are there in the tomato. She needs to compare the expression with pepper. And then those genes, exactly like in pepper, you modulate the expression. And then the tomato will have the pungency genes. So if you want to make salsa that time, you can just use these genetically modified tomatoes that can make pungency also, right? That is a possibility, a theoretical possibility. So this is the chapter 5. So now we will discuss chapter 6. Chapter 14. Why we are switching from chapter 5 to 14? In the midterm 1, that is the first midterm, I included these first 5 chapters and chapter 14 and chapter 15. Chapter 14 and chapter 15 are related to basic chapters. So why to have them at the end? So that we will have all the 7 chapters for the test. So we have been talking about molecular evolution, right? So how the speciation occurs, how the chromosomes will get duplications, some of the domains get duplications, or some of them will get deletions. So what is the underlying mechanism? That is, during the meiosis, crossover happens, and crossover can be unequal crossover, making some chromosomes gain something, lose something, which will eventually form into different species. And since they are moving to different place, they express differently. Like chromosome, one part you put it in the chromosome 14 or chromosome 13, that will express differently, right? You don't speak exactly the same way when you are out of country. Say you are totally different place, you are going to Alabama, you try to modulate your accent. You try to be like Alabama kind of guy, that accent. You don't talk like in West Virginia. So that is the position effect. So once the genes are moving from one chromosome to different chromosome, those genes will behave differently, express differently, making it eventually different species. So we need to understand the molecular evolution and the population genetics. So what is population genetics? How at population level genes behave, not single genes. Like the genes located within you or me do not matter. But as a subpopulation, it matters. As a population, that is diverse population here. I'm from India, and then somebody is from Africa, somebody is from Nepal, Mexico, US, with various European descents. So our classroom is diverse. So we are a population. And then this population goes to some place, migrates, just like all of us ended up in Charleston. We become subpopulations. So the genes that are there, the population level genes, and then once it is subpopulations, what kind of evolutionary forces occur on these genes? How they become diverged from the original population? Say somebody is from Africa. If they are here in the US and African Americans, their genome is quite different because they have admixed with the local lineages, right? So you have complex genomes. Or within, from different parts of Africa, people came and then admixed with each other. And the European lineages, African lineages admixed to produce a subpopulation. This subpopulation is different from the original population. You see, the original populations, I do not know, did I switch on the AI? I'm not sure this class. Okay, we will figure out. Probably it is all happening. So we need to understand what is the evolutionary relationship between the species. So we talked about populations, human populations and subpopulations. We are not the only species in the world. There are so many different species. We have biodiversity. We have animal species, fungal species, bacterial species. Okay, so many kinds of plant species. So they have the populations. Insect population in West Virginia. Probably you never met a particular population of insects that are dwelling in West Virginia. Maybe much more, thousands of years. You cannot say you are more profound West Virginian. They are probably here for thousands of years. Very native plants, very native insects, very native birds, very native fungi. The mushrooms you see, West Virginian mushrooms are so diverse. Go to the wilderness and collect various mushrooms. You can see very, very colorful mushrooms. So these are all native species. See, these native species will have populations. But if the population is migrated to a different place, what will happen to that population? That population will get admixed with other populations, other individuals of the population, and form subpopulations. So these subpopulations diverge. If it is plant species, the subpopulation that is formed in West Virginia, for example, if it is invasive, coming from China or India or any other Asian country and getting distributed here, and a subpopulation is forming, eventually these subpopulations will become ecotypes. You see, suitable for an environment, one particular environment. So they are existing for a long time in these hills, mountains, where the climatic conditions and adaptive features are different comparatively from where they have migrated. Okay, but humans migrated to the United States pretty recently, or probably the natives existed in the United States were never properly admixed. Probably they did to some extent, but not clearly admixed with European or African populations, I suppose. So our admixture history of humans living in the United States is not very wide. We are pretty history of 500, 600 years here. But the plant species or the animal species, insect species or aquatic species, they are all here for several thousands of years. And some of them probably migrated during various geographical changes that is happening. Or migration might have been by any way, just like oceanic currents are taking African cottonseed to Mexico when there was no humanity around. So they are undergoing their own evolution, okay? And then once the subpopulations are formed, these subpopulations are diverged from the main populations because they are adapted to a particular environment, they become ecotypes. You are an ecotype of West Virginia. If I am putting you in San Diego for a couple of days, you feel shock. Hey, what is this? Where am I? How to do it? Or Florida, for example, altogether geographical region. You never went there, altogether you are dropped there. And how you are going to face a different environment? So you are kind of ecotype. But you within ecotypes are not completely diverged. You can find somebody there, you tell, hey, I don't know anyone here. Can you help me to get around? He or she will drive you around and show things. This is how you do in Florida. You don't do these kind of things in Florida or a different place. Then by end of the week, you will learn what to do, what not to do. Because ecotypes are not genetically diverged. But if you are genetically diverged, then what happens to you? Then you will have wide differences genetically. Then there will be compatibility issues. Sexually, you will become incompatible. Don't think in terms of humans. Think about insects. Think about aquatic species or birds. When the subpopulations diverge, they become ecotypes. When ecotypes diverge, they become species. Once they become species, they will not. They will be reproductive barriers to mate. You see, this species is different. This species is different, and then these two species will never reproduce the way they were. Right? So the species also diverge, and become what? Genera, genus. Like tomato is a genus, pepper is a genus. Once upon a time, they were same species, different species. So a simple single population can become subpopulations. Subpopulations can become ecotypes. Ecotypes can become species. Species become genera, and genus become different families even, totally. You are totally different family. Like sweet potato and Solanaceae are very close. But sweet potato is totally different from all this. But initially, several millions of years back, they had a common ancestor, and sweet potato and Solanaceae vegetables probably had a common ancestor. So by doing DNA sequence analysis, you can understand this evolution at molecular level. And then you can build relationships using these methods that are developed called comparative genomics. You can develop methods to identify evolutionary relationships among the species. So that field is called molecular phylogenetics. By to understand the differences, for example, human species, we are not the only species. We are the only species probably, what, 30,000 or 40,000 years. Before 40 to 50,000 years, there were several other human species we call archaic hominids. So what are those hominids? Some of them were Neanderthals, some of them are Denisovans. Okay. So all these different humans came to either closer to Europe or Middle Europe or North Europe, and eventually some of the species mixed up. So the modern human, what we have here in the classroom, we are all modern humans. We are not of one single species. We are mix of various human species. But if somebody is coming originally from Africa, they are pure Homo sapiens. Okay? So if you are admixed with the other lineages, the Indians are not Homo sapiens. We have a mixture of Neanderthal genome. Chinese also have a mixture of Neanderthal and Denisovan genome. And East Europeans are mix of same. So similarly, the human race is formed because we have all different species have undergone admixture. Exchange of the lineages happens. So using this kind of molecular evolution, DNA sequence data, we can understand what is the extent of divergence. For example, we all know that we all evolved from the chimpanzee. Okay? You cannot refute that. You cannot say that, no, I am not from chimpanzee. My parents were royal. They were kind of landlords. No, everybody eventually, if you trace back our lineage history, we are all related to chimpanzee. And the genetic distance is just 1% between the DNA of chimpanzees and humans. Then with just 1%, why chimpanzee is in forest? And why are we here in city driving cars and listening to the genetic lectures? Because our genes express differently from the chimpanzees. Chimpanzees cannot speak. but they have the genes that can make them speak. But the way they express, the way they produce different amounts of proteins, and the mix of that amounts of proteins will form different phenotypes. One became human, and other became chimpanzee. Within human, we are different races. All these are formed. For everything, we can estimate a molecular evolutionary distances or phylogenies. So similarly, the gorilla is the next ancestor. So at some point here, the gorilla diverged into this cluster called human and chimpanzee cluster. Okay. And then who is ancestor for the gorilla? Orangutan. Okay. Now, orangutan is genetically quite distant. But when the orangutan is placed closer to chimpanzee and gorilla, it can share more features with gorilla rather than the chimpanzee. And human is way distanced. And then the baboon, which is the most ancestor for all these primates. So this kind of evolutionary relationships can be formed. Okay. And then within humans, say 1% is the difference between human and chimpanzee. Now we are 99.9% identical, all the humans. Though we look slightly different from each other, our skin may look different or our heights may different. The way we sound is different. That is all within this 99.9%. So that 0.1% of DNA, that makes it difference because the genes express differently. The regulatory domains that are there, regulatory switches for each gene will alter. With those slight alterations are good enough to cause enormous phenotypic diversity. So you can build gene trees based on one single gene. You can build a tree and say this is a gene tree. And also you can build a species tree. For making species tree, you do whole genome sequencing and build a species tree. So with one single gene based on that you are making a tree, that will not explain correct relationship. Okay, they look different. For each gene, it is different. For example, John is different here in the classroom, but John is different in different place. Maybe he is an artist, or maybe he is a football player. So when you take the John's behavior in different places, he is different in each. So for each gene, the species are different. For example, how the relationship. Maybe when he is playing football, his relationship is close to the other players. But if he is sitting in the classroom, he is a geneticist, listening to the genetics, talking genetics. So he is more closer to the genetics. So when you take just one situation and relate, that is not going to be universal feature. If you want to understand John, you need to go with him together for several months. What he does, what he eats, how he behaves, how angry he can get, how fun he is, how gentle soul he is, or how aggressive he is. And then totality, you can build a tree, and that is more robust. Similarly, the species trees are made from the whole genome sequence. The entire chromosomes will be sequenced and build trees. And they are anyway different from just one single gene tree. Here is an example. So here is a tree. There are three species: species 1, species 2, and species 3. Okay. So species wise, species 1 and species 2 are together. They are the first species. And then species 2 and species 3 are together. So species 1 is distance from the species 3. That is at the species level. But one gene, if you think, that is the gene A, that gene A is totally initially just one gene, and then that undergoes mutations, and those mutations will cause it a different form of the same gene. So this is different allele, okay, A2 allele. So this A2 allele, A1 allele, there are two alleles. So in one species, it could be end up A2 allele. In another species, it could end up A2 allele. And in one species, it can be in A1 allele. So based on that, we cannot say, hey, these two species are together, but they share the same alleles. I'm sorry to say species 1 is different from species 2 to species 3. Species trees will say that species 2 and species 3 are together, and then they are diverged at this point from the species 1. Okay? So this gene, which is initially 1, at some point it got mutation and a different phenotype. And then that gene is traveling through the evolutionary distance. In species 1, finally the A2 is fixed. A1 is lost. A2 is fixed. Okay? But species 2, which is whole genome level, it is closer to species 3. Species 2 and species 3 are close. But just for that gene, this A1 gene converted to A2 in species 2. So A2 gene is shared between species 1 and species 2. Based on that, you cannot say, hey, these two species are very close. It's only one single gene. It's not, we have 30,000 genes, for example, in humans. So many differences. So you cannot say, just based on the one gene, myself and John are brothers. We may not be. We are not, apparently. But for one gene, it is possible John and I can share a particular same allele just for speaking purpose, just for thinking. But species 2 and species 3, for this gene, this has A1 gene, this has A2 gene, which are different. But these two species as a whole, they are similar. Okay, though species 2 and species 1, they share A2 genes. So we can understand sequence evolution. We can say, well, the dinosaurs were here, and modern chicken is the descendant of the Tyrannosaurus. So it can say, the chicken can say, how dare you eat me? My ancestors several millions of years ago can eat 10 humans together, or 20 humans, they can bundle up and chew together. That was such a glorious history. But the Tyrannosaurus genome evolved, and the closest genome what we have is the chicken. Okay, not even elephant. So any divergence can be estimated. Any existing species you can estimate. We have rice, maize, sugarcane, sorghum, foxtail millet, or tomato, peppers, sweet potatoes. We have diversity in plants. If we want, we can sequence and estimate which is closer to what. What is the closer to cotton? Can you imagine what is close to the cotton? Grapes. We published that paper in the Nature. That is my postdoc work. You go to my Google Scholar and see the Nature paper. In that paper, we published that cotton is close to the grapes, and grapes is closer to the chocolate, that is cacao. Okay, that is the paper we published long time back in Nature. Okay, so is it not, can you guess that? If I say which is closer to cotton, can you say grapes are closer? And then chocolate is closer to grapes? It's very difficult to understand by looking at the phenotypes. You cannot guess this chicken, which is, you can buy a cup of nuggets, a pack of nuggets from the McDonald's, had a huge ancestor that is Tyrannosaurus, right? So, but you can estimate that, you can sequence all these organisms. Fossils you can sequence, or the existing or extant genomes you can sequence, and then you can do this molecular clock. you can estimate how the constant rate of change has been occurred. There are tools, guys, to understand how these molecular clocks evolved. And this long periods of evolutionary time, that has changed. The rate of amino acids are changed in the gene. The gene is nothing but the codons, triplet codons. Once gene is transcribed, it becomes what? Messenger RNA. And messenger RNA will have lot of triplet codons, and each codon is an amino acid. So you are seeing how these amino acids are replaced over the long period of evolutionary time, using which you can estimate molecular time, how much time it took, what kind of evolutionary perturbances chicken faced when it is evolving from Tyrannosaurus to chicken, or how the grapes became cotton, or how chocolate, which should ended up in nice chocolate drink, has evolved into cotton, grapes. It's all unimaginable. But this evolution will help you to understand the molecular clocks, how to estimate molecular clocks, and what might have undergone. So let us slightly switch the gears. So there are mutations. So a sequence will undergo mutations. So what is a mutation? One nucleotide is substituted by another nucleotide. And how many nucleotides we have? 4. Adenine, guanine, cytosine, and thymine, we have 4 bases, and then 4 nucleosides. And once the base is combined with both sugar and phosphate, it becomes nucleotide. So if it is in DNA, it is deoxyribonucleotide. If it is an RNA, it is ribonucleotide, which is okay. And the deoxyribonucleotides, once in DNA, they get transcribed and produce messenger RNA. And that messenger RNA is translated into proteins. So on the messenger RNA, you have triplet codons, a codon after the codon. So these codons are very important to be translated, because the tRNA, transfer RNAs, can look at these codons and bring appropriate amino acid to the place. You see, here is a messenger RNA with multiple triplet codons. So each codon is specific to an amino acid. Who can read that? Somebody needs to translate it. So there is an RNA called tRNA, transfer RNA, that can bring the appropriate amino acid to the particular codon. And all these amino acids are assembled into polypeptides, into peptide chains. So what is in a peptide chain? Amino acids. And why they are assembled in that fashion? Because each amino acid has its own codon. And where are these codons? In the messenger RNA. From where did this messenger RNA came from? The DNA. DNA got transcribed into messenger RNA, and messenger RNA has bunch of these codons, and these codons are transformed into polypeptide chains. These polypeptide chains are folded into proteins. Okay, so naturally, if a nucleotide is changing, a codon is changing, right? And then there will be different amino acid in that protein. Because it has different amino acid in the protein, that can behave totally different way, because it has different chemical form. The way that polypeptide chain, having different set of amino acids here and there, can fold it in different fashion. Okay, so its functionality is lost or reduced. Then it will form a different allele. Okay, having this protein, the guy has blue eyes. Having this protein, the guy makes pigment which is brown eyes, or brown color skin, or black color skin, or yellow color skin, or different any color of the skin, whatever we see in humans. So similarly in the birds, similarly in the insects. In the dogs, you see, there are huge dogs, two very small miniature dogs. So much differences because they all have different kinds of proteins. And from where are the resulting different forms of proteins? Because they have undergone mutations. And those mutations led to produce different kinds of proteins. Okay? Can you expect by mating two big German shepherds, you can never get a different kind of dog from there, a smaller feature dog like Chihuahua. You cannot get it. You will, when you mate two German shepherds, always babies are German shepherd. Because German shepherd genes will transcribe a messenger RNA. That messenger RNA is going to get translated a particular protein, and that protein is important for the phenotype, what we look as German shepherd. Okay, but Chihuahua has a different forms of protein. Same homologous proteins are there, similar proteins. Why did it happen? Because at some point, DNA sequence has undergone mutations, and those mutations are causing those changes. Now, here is the most important topic. You know, there are 4 nucleotides, adenine, guanine, cytosine, and thymine. If you want to make triplet codons, 3 different codons, so how many nucleotides you need to take? You need to take 3 nucleotides at a time. Okay, then there are 20 amino acids in the nature, whether you are plant or animal, and bacteria or fungus, whoever you are. Okay, you work with only these 20 amino acids. And these 20 amino acids have a code. But then how 4 nucleotides can make codons for 20 amino acids? If you think that the codon just has 2 nucleotides, then 4 times 4, you can only explain 16 amino acids can form, right? So it has to be triplet codons, 4 times 4 times 4. That is 4 times 4 is 16. 16 times 4 is 64. So you will make 64 codons. And there are 3 stop codons. You will understand what are the stop codons in future. And there has to be one start codon. So rest of the 60 codons need to make these 20 amino acids. So there are synonymous codons. So same codons can produce same amino acid. I mean, 3 different or 4 different codons can produce same amino acid. They are called synonymous codons. So a mutation is occurring, but that mutation is not changing the amino acid, but nucleotide change is there, but amino acid is not altered. They are called synonymous substitutions. What are the synonymous words in English? Stupid or idiot. Both are same. If somebody you are calling stupid or idiot, that means you get the meaning that the guy is cussing me, right? So sorry for bad words. So that means what? Similarly, for same amino acids, there are different codons. So one codon is altering to the other, but still if it is synonymous alteration, synonymous substitution, it is not going to alter the amino acid. If the amino acid is not altered, finally the protein will be the same. Okay, so synonymous substitutions can happen many times because that is not altering your protein. You see, you never can produce a human with a dog face. Can you produce? You cannot. Why? Because we will not mutate that drastically, right? Can you think that you will produce a Godzilla or an elephant as baby? No. As long as a human you produce a baby, that is going to be a human. Why? Because we will not mutate. Our mutation boundaries are similar. But the dogs, chimpanzees, humans, we all have same genes. Then why don't we alter and drastically and produce an elephant head or a monkey head? We will not, because our mutation boundary will not exceed a limit. And then whatever the mutations are happening, they are not changing the amino acid. They are just changing the codon. So what is happening? majority of our mutations are synonymous substitutions. But we cannot say all the time. Within humans, some of us get cancers, some of us will not. Some of us are small, some of us are large. Some of us can play really well. We have more agility for playing games. And then some of us are kind of very docile, just cannot play. We are always sick and tired, like me. So anything can happen like that. So this still some mutations do happen, and this mutation load persists in the population, right? So a mutation load specific to African Americans, a mutation load specific to all humans, or mutation load specific to Chinese, mutation load specific to each subpopulation, Indians or Germans, Irish people. That is subpopulation specific mutation load. But as a whole, also human mutation load. You cannot exceed that. The genome will not undergo that kind of perturbation that you produce a baby like a lizard or totally like a plant, right? So, but most of the genes, especially the animal genes, are similar. And chimpanzee and we share almost 99% of the genes. But why don't we see, at least in world, one chimpanzee baby coming from the humans, or vice versa? Chimpanzees are producing a human baby. It is not possible because we have boundaries already set. Our mutations will not exceed more. But within the boundary, one chimpanzee is different from other chimpanzee. And similarly, one human is different from other human because of non-synonymous substitutions or the amino acid changes that happen within those boundaries, making us different. So we have mutations, synonymous mutations that do not alter amino acid and keep our species integrity. And also there are non-synonymous substitutions. If we are not choosing the right partner and producing a baby, it can be a sick baby, all the time sneezing and coughing, right? Or very intelligent baby like Einstein. So you have the power, you have the right to choose your partner to produce a baby. That is fitness. We will understand that fitness later. What is fitness? So now the genes we are talking, the genes that are there in different species, like genes, same gene in the chimpanzee and human is exactly the same gene structurally. It will produce same protein, but the protein amount differs because that gene is expressing more, this gene is expressing less. So the genes structurally are diverse. You can identify, oh, this is the blood of cow, not human blood. Like somebody killed someone and poured their cow blood. The forensic scientist will identify, hey, this is not human blood, this is cow blood, or this is goat blood, or monkey blood, or dog blood. How he can identify when we have the same blood of globin genes, same hemoglobin, alpha and beta hemoglobin, tetramers are there in the blood, but still those genes are structurally different. Those proteins are different. So you can identify that. So the genes, say, since we talked about the blood, globin genes are important for making the blood. Alpha globin and beta globin, there are two different globin genes. So two proteins of alpha globin, two proteins of beta globin can make a tetramer. That is one single cell, hemoglobin cell. So the genes, globin genes of various mammals are orthologous to each other. Okay, the cow has, goat has, all mammals have blood, produce the blood. Are reptiles, not reptiles, birds will produce blood. So you can understand how those genes are assembled, how genetically diverse they are, because they are orthologous to each other. Okay. The genes that are in various species that is accompanying with speciation, but retain the same function. They are still producing the blood in goat or cow or chicken. Function is the same, and blood does the same job in the chicken also, in the cow also, in camel or elephant, wherever it is, the blood function is the same. But the genes are different. They make slightly different protein, though the function is the same. They are called orthologous genes. And then there are the paralogous genes. Paralogous genes are within same species. So like you have alpha genes and beta genes, beta globin genes. So you need both alpha globin genes and beta globin genes. Both are needed to produce a complete hemoglobin. So that means what? Though these genes had a common ancestor, once upon a time, the ancestral mammal, our ancestor to mammal that started making the blood is having same gene, and then that gene is duplicated. So one gene become alpha and another gene become beta, and both these genes are needed to produce the same function. Okay, so within species differences, same species or the same organism, the duplicated genes are called paralogous. So why this is all happening? Why this is all happening? So somebody put some point here. Okay. So why this is happening? Because genes are getting duplicated or triplicated. So how the genes are getting duplicated and triplicated? Unequal crossover. Okay. So same process is duplicating or triplicating. And then once the genes are diverged, like one particular gene is there, and once it became doubled, what happens? It will start accumulating mutations differently, right? So one will act differently, the other act differently, and then they have two different functions, and collectively they can do something together, like making the hemoglobin. Like globin gene was initially one that is duplicated because of unequal crossover. And then that duplication resulted into alpha and beta. And then alpha started making different protein, beta started making different protein, and collectively it can make a hemoglobin. Okay. So, and then during that process, some of the genes will be mutated so drastically and they will lose the function. Now you know that this is a pseudogene. Pseudogene do not have any function. They cannot transcribe and produce a functional protein. They lost the function. You can identify, oh, here is a pseudogene. Okay. So you are traveling somewhere in the highway, you can find a dilapidated house which is 300, 400 years back, and nobody enters in there. It is not functional. Like that, you can find pseudogenes in the genome. Once you sequence the genome, you can profile the genes. You can understand what are the genes that are functional and how they are different with various species. That is orthologous. Pseudogenes are the silent genes. So you can understand the ancestral gene and how this ancestral gene undergoes duplications and how the mutations accumulate, resulting in paralogs, and then how some of the paralogs can become a pseudogene, and alternatively, one paralog sometimes can acquire new function or pattern of expression, right? So a gene can differ in its expression. One can express like ancestral gene, and the other one can reduce the expression or increase the expression or completely produce a new function. Like I am duplicating you. For example, berry is duplicated into 2% using some genetic techniques. Then what would these two berries will do to each other? Whether they will be friends and they will say, hey, you go and work in store, I will go to the class. Or you go to party and I just do the homework. So the duplicated humans, they are the same. That means they are synergistic to each other. They are friends to each other, right? But one berry gets angry on the other berry and he says, okay, let me tie you and put you in the attic. You are causing me a lot of trouble, okay? And then that becomes like pseudogene. Or they can say, well, let us both study. Let us go, you go to organic chemistry, I go to genetics class. That is wonderful. That is all kind of synergy. Sometimes new function. They say, hey, you go and make money totally differently. You play music, I go to science class, attending new function. So once there are duplicated genes, these duplicated genes can work in the same direction, producing the same thing or diverse things. That's what I'm trying to say. Or becoming totally pseudogene, that one gene totally is inactivated and only one gene remains. So here is a thing, a globin gene is there. This globin gene is duplicated. And once it is duplicated, it is diverged. And this diverged protein is slightly different. One is called alpha and another is beta. And then they were initially on the same chromosome, but transposable elements are another evolutionary force. They can move this gene to different chromosome, to different chromosome. So now this alpha gene, which is on different chromosome, is on chromosome 16. And this beta gene, which moved to different chromosome, is on chromosome 11. So they started on different chromosomes. But once upon a time, during the evolutionary time, they were together. And even before becoming together, they were exactly same copies of the same gene. And initially, it was an ancestral gene, one gene. And then this alpha gene became zeta and alpha. This zeta gene is also important, and it works very nicely in the fetal stage. When humans are fetal or embryonic stage, you need zeta form of alpha, okay? And then this signal is for pseudo. So zeta became pseudo here. Alpha 2 is pseudo here. Alpha 1 is pseudo here. And then alpha 1 and alpha 2 are functional genes here. So alpha gene is also two kinds, alpha 2 and alpha 1. So all these genes, these pseudo genes and functional genes that are there on the chromosome 16 as a cluster is called alpha globin gene family. Okay. And the beta also has undergone duplicated, diverged to form epsilon gene and gamma gene. And then this is the beta gene. So this epsilon remained epsilon. This gamma became gamma G, gamma A, in the sense just different for one nucleotide at one position. So still we call it as gamma G and gamma A. And then this is the pseudogene of gamma B. And then this is another gene altogether, delta, okay, and beta. All this is the beta family. So all the genes collectively in different tissues, in different developmental stages, they alter their function and collaborate to form one molecule that is hemoglobin. You can say this is the fetal blood. This is embryonic blood. You know, this is liver is damaged, 4 and 6 you can see. Because based on this proteins, you can identify how you got the blood using the 4 and 6. Any questions, folks, so far? Are you all around and sleeping? When to sleep? Any questions, please type in chat for a couple of minutes. Sam says no questions. That's nice. Kelly, Cheston, everyone saying no questions. So looks like very fun. Okay. Any questions, anytime we can meet. And John also says no questions, though he was cited multiple times during the class. Justin. So everyone is very comfortable with the class. Glad to hear that. Okay. Otherwise, we should discuss again. We have the whole semester to understand some genetics. Okay, now we understood already population, which is individuals of species living in the geographical area is a population. African American population or Caucasian population, and subpopulation is say African American population of Alabama or New York, or subpopulation is say you can say Colorado or West Virginia. Subpopulations, local populations are within West Virginia, Charleston or Morgantown or Clarksburg, Huntington, local populations. So what happens, why this is important? Because the populations, subpopulations already formed, their migration is restricted. How many days, how many times you travel to Colorado? Probably once in lifetime, just as a tourist. Otherwise, you are a West Virginian living here, or you might migrate also, totally. And once subpopulations are distinctly formed, especially in insects or animals or aquatic creatures, their migration is restricted. Okay. So there may be geographical barriers. For example, we have two lakes that are distanced by, say, 500 miles, Lake 1 and Lake 2. So aquatic species of one lake and other lake, they cannot migrate. There are barriers. And then probably those two lakes were in the uh, in history. Historically, it was one single lake once upon a time and became two lakes, and the populations or species or biodiversity is different. Anything you can imagine. So now the gene pool is the collective genetic information of the individuals within that population. It's the gene pool. And you can estimate genotype frequency. How many are dominant genotypes? How many are heterozygous genotypes? How many are recessive genotypes? You can estimate. For example, we want to estimate for a gene within the Charleston. Charleston has probably lot of Caucasians and African Americans and some Chinese people, some Indian people. You can come across various races here and there in Charleston city. But if you want to estimate genotype frequency, what do? You take some samples from all the people and see how are the dominant homozygotes, how are the recessive homozygotes or heterozygotes are distributed in that population. And then you can count the alleles. Homozygotes will have two alleles, heterozygotes will have one allele. So you can also estimate the allele frequencies. So allele frequencies are estimated from the genotype frequencies. Okay. If a seed is capital W, that means that is round for homozygosity for round. And capital W, small w is a heterozygous, and small w is a recessive. So if you need to count the alleles, you can count. Heterozygote has one each, one capital W, one small w, and some small w. Like that, you can estimate the allele frequencies. Yeah, very good. So here is a very funny locus. This is actually data from France. In the book, you have the story. So this locus is called chemokine receptor on the T cells. On T cell surfaces, immune cells, T cells, thymus cells, which makes antibodies, right, to prevent any infection. They have a receptor called chemokine receptor. So once you get the HIV virus entry, HIV, that virus entry, that can enter, that virus-specific RNA comes to your body, into your genome, into your nucleus, and then it becomes double-stranded. And then through this reverse transcriptase, and then that can get integrated into the chromosomes. And since then, that time, you start making your genome or your chromosomes will start making virus-specific RNA, and that would be keep on integrating. The cycle continues, retrovirus. So this virus can enter into cells, especially immune cells. Till then, it's not a problem. You are just HIV positive. You will never be AIDS patient. When you will become AIDS patient, because this HIV virus makes entry into your T cells or B cells, immune cells. So how they can enter into those cells? This virus can only enter through a receptor. There has to be an entry. That entry is called chemokine receptor. It's a protein. Okay. So if somebody has a deletion for that chemokine receptor, a 35 base pair deletion can happen. And then that person is immune to HIV virus. HIV virus can be there anywhere in the body, keep on integrating into the genome, but it will never enter into that person's immune cells. So he or she will never become AIDS patient. Okay, they will be HIV positive throughout the life because they have 35 base pair deletion in their allele. That's why they cannot make chemokine receptor. If that receptor is not there, HIV virus can never enter into cells. So the normal people in France, they took the data of 1,000 people. These 1,000 people had 795 people had capital A genotype. In the sense, they will make perfect chemokine receptor. And then they can get infected with HIV and become AIDS patients because there is a receptor, and then the virus can make into their genomes and convert them into AIDS patients. So if it is 795 A capital A genotypes, you can do the PCR by designing primers for this chemokine receptor and then understand how many genotypes are there. Then you will see 1,590 A alleles because 795 AA genotypes. That means there are two dominant alleles here. 2 times 795 is 1590 capital A alleles. And then lucky people who are having that mutation that will never get AIDS in spite of their HIV positive are just 15 people out of 1,000. So 15 in the sense that is small a genotypes. There are only 30. 15 times 2, 30. And then there are this 190 people that are heterozygotes. So heterozygotes will have capital A, small a. So 190 of them are capital A alleles, 190 of them are small a alleles. Okay. So now this number of alleles, 1,000 people, 1,000 times 2, that is 1780 of capital A alleles here, and 220 of them are small a alleles because 190 plus 30, 220. 190 plus 1590 is 1780. The total number of alleles is 2000. So what is the frequency of A allele? If you consider frequency of A is P, 1590 plus 190, that is 1590 is here, 190 is in the heterozygote. That is divided by 2000, that is 0.89. 89% of the alleles in that 1000 people are capital A alleles, and small a alleles are Q. That is 30 plus 190. 30 are recessive, so 15 are individuals, because each one of them have 2 alleles. 15 times 2, 30 alleles. 30 alleles plus 190, that is 11%. So P is estimated like this: 2 times of capital A, plus half of capital A small a divided by 2n. And Q is 1 minus P. Once you estimate P, you can estimate Q easily, because out of totally, you say here, if I give you 0.89, and you know how to estimate frequency of A, you don't have to go through all this. You will say 100 minus 0.89, so 11 is the frequency of A. So Q is naturally 1 minus P. Why this is important? In the sense, for example, there is a subject called epidemiology. What epidemiologists will do is estimation. Say you say one cystic fibrosis. Cystic fibrosis is a kind of lung disease that happens to only white people. One baby happens for every 1,500 people. Cystic fibrosis. And then you can estimate what is Q. This is Q. You can estimate now 1 out of 1,500, you can estimate what is Q here, frequency of that recessive allele, because 1 is occurring at every 1,000. And once you know the Q, you can estimate P, like what is P? 1 minus P equal to Q. So once you know Q, you know 1 minus P. So if you know P and Q, now you know P, you know Q, just because of one data, that is hospital recorded, that in Charleston, there is one cystic fibrosis baby for every 1,500 babies born. That means you can estimate PQ, which is heterozygosity, because most of the humans are heterozygous. So now you can say, based on this frequency, you can say, based on the PQ, you can say, well, every one individual in every 21 adults is heterozygous, is a carrier. Okay. So for example, one in every 21 adults can carry cystic fibrosis allele, causing 1,500 out of 1,500 randomly born babies, one will be with cystic fibrosis. Okay. In different places, it could be different. For example, you are going to a different city, Albuquerque in New Mexico. And then there could be, you ask that big hospital where maternity hospital, they will say, hey, we get one cystic fibrosis patient for every 500 babies. That means what? There are more heterozygotes in that area, in that city, that are carrying this allele. That's why there is more frequency of babies getting birth for every 1,500 births. Okay. So there will be more heterozygotes. Then there should be more lung doctors, right? More facilities to cure them, more medicines to cure cystic fibrosis. So this field is epidemiology. So epidemiologist's job is to estimate the disease risk in a particular area. So otherwise, if there is not much risk, you don't recruit that lung specialist, that machinery, and that pharmacists will not have the drugs related to that situation. All those things you can minimize. But in a place where lot of heterozygotes are there, lot of couriers are there, then you need to have more facilities to, because you know that more babies are going to born with cystic fibrosis. So that's how this data is very important for epidemiology. So now you have genotypes, allele frequencies can be estimated among the reproducible adults. Say out of 15, that means the genotype frequency, what is it I'm talking? Yeah, genotype frequency is 11 and 89.89 capital A. This is coming because half of heterozygotes are coming from here, and then half of, I mean, capital A is coming 0.795, and this one is added. So the same thing what I discussed earlier slide, showing it differently. How you can estimate genotype frequency and gamete, this allele frequencies. So the estimation of this is very important to understand the outbreaks, very practical application of it. Then also you can understand how this evolutionary forces is operating. If a particular allele is more preferred, say the heterozygotes are more preferred, then there will be increased number of heterozygotes comparatively to dominant and recessive alleles. Or if recessive allele is more important, then eventually in the population, recessive allele is going to get fixed, and there will be more individuals with the recessive alleles, homozygous recessive alleles. So if there is always a dominant allele, which is having some effect, phenotypic effect, it can cause a disease or it can be beneficial. Either way, it is going to spread in the population very fast because the dominant alleles will have higher frequencies. We will understand that. So we can understand, we can say that this capital A, small a, is in this equilibrium, p squared 2pq and q square equilibrium. If this equilibrium is equal to 1, that means what? That means the particular locus is under equilibrium, Hardy-Weinberg equilibrium, and that is not associated to any phenotype, or it is not getting selected. If it is not getting selected in the sense, here you are saying 0.89 and 0.11 is becoming exactly 1. That means though the French people may get AIDS or not, but nobody is selecting. No Frenchman is looking for a mate and understanding whether the female has what kind of allele before he produces babies. That do not happen. So as long as they are French, probably they can speak in French, they can meet and produce babies. That is what primary force. Not that a Frenchman will ask a woman, hey, what kind of alleles you have for the chemokine receptor? Let me decide to have a baby with you or not. That is not a practical way of reproducing in humans. So these allele frequencies are maintained because there is always a therapeutic intervention for AIDS also or HIV also. If somebody is HIV, he will get tested and then he will use retroviral drugs so that the infection into the immune cells is contained, whether he has whatever locus he is. So therapeutic interventions can keep the balances constant. So if there is no therapeutic, there is no medicine for AIDS, what can happen? Like then that can become a scary thing that, just they are having a unprotected intercourse, then the people will get AIDS, HIV, and then all of them are dying. There is no intervention. And then what happens? Then the homozygotes and heterozygotes will reduce, and only the people who can survive effectively is with the people with the deletion in the chemokine receptor. Okay, That is not the case in the case of this mutation because there is therapeutic intervention. People, even the HIV, can live longer. So all the genotypes can exist. That's why the frequency of capital A, frequency of small a is equal to 1. That means it is in the equilibrium. When this equilibrium will dwindle, is a particular genotype is favored. If a heterozygote is favored, then you will see more heterozygotes because homozygotes are not surviving. And then we have only heterozygotes. So you can understand the human loci in the population, whether they are in Hardy-Weinberg equilibrium or they are kind of dwindling towards recessive or dominance or heterozygosity. So the female gametes will have, similarly females also can produce capital A, small a, and males also can produce capital A, small a. So as long as random mating is happening, then the frequency has to be p squared to 2pq to q squared. That means there is no selection. So when the selection will happen, when any of these genotypes will start dying for a condition, they cannot withstand that perturbation when they are dying. Like if there is no medicine for HIV, no retroviral medicine, probably in all the populations, only these people will survive. If, say, for example, retroviral, this HIV infection can happen with just handshake, then what happens? There is there is no medicine. There is no drug. And then what can happen? How people can prevent it? No way they can prevent it. Only one genotype can prevail. Rest of the people will die. Because these people will prevail because they don't make chemokine receptor and HIV cannot enter into their cells. So they will never become AIDS patients. But such is not the case. There is intervention. That's why natural balance is maintained. That's why we say we can estimate p square plus 2pq plus q square for understanding what is the frequency of a particular genotype in the population. And if we know what is the function of that allele, then we can understand how the epidemics of that particular locus is working in that locality or in that subpopulation. So in ideal population, that means if a population is there and this population randomly mates, not in humans. Let us think about insects or birds or aquatic species. The population is large and random mating is happening. Each fish has equal chance to mate other fish or other bird. A bird has equal opportunity to mate the other bird. Similarly, the insects. And then what happens? And then allele frequencies or genotype frequencies, they remain constant. There is no perturbation. But if any bird flu happens or any kind of aquatic disease comes, and if a particular genotype is favored, then rest of the genotypes will die. Okay, so there is natural selection there. So if there is no such selection, if the population is large, then there is no perturbance. Happily, the allele frequencies and genotype frequencies will remain constant in every generation. Now or in the past, in the future, they will remain constantly same because there is no perturbation, there is no selection. But the natural selection will happen for all these things. New birds will come into the area and will start eating insects. Or the insects will start become mutant, more virulent, and start eating crops. So one way or other, everybody, all the organisms, we are all subjected to selection. Okay, that's what Darwin said. What Darwin said, what is the most evolutionary force Why are we evolving? Why are we mutating, accumulating mutations? Why are we migrating? Everything is the reason for adaptation, for survival. Why some birds move to West Virginia? Why some insects move out of West Virginia? Or why some invasive species come here and populate, become large? This is all because of survival, isn't it? And once they are migrated, what evolutionary forces will accompany them? They need to adapt. If they are coming to US, you need to adapt US things, US style. You see, your ancestors, Europeans, when they came here, they eventually adapted, right? Initially, they were not adapted. They've undergone a lot of troubles. So that's why we celebrate Thanksgiving, because some of your ancestors could not survive by themselves and they were getting supplies from natives, something like that. So once you migrate, why do you migrate? For better life, for survival. Why did your ancestors came from Europe to US? For better life. And then what was the important thing immediately they have undergone is adaptation. So once they adapted, then they made it home because they know how to get around, how to cultivate here, how to form a governance, and how to have a law, and things like that. So any population, any species that is undergoing a migration, that migration has to follow with the adaptation. And similarly, the mutations will happen. So a new mutation is occurring. Then what is important is that mutation, whether the newly acquired mutation is helpful or not helpful. If it is helpful, that get fixed in the population. That too, if it is a dominant allele, then that can spread very fast. If it is a recessive allele, because the proportion of recessive alleles in the segregating populations is less, it takes longer time to get it fixed in the populations. So, what is an ideal population? Ideal population is there is no selection. There are no diseases at all. You don't have to go to hospitals. You don't have to worry about food. Every time you will get healthy food, and all the time you are physically fit, healthy. Then there is no selection. And then if everything is working for you, you don't have to work hard. You just nicely live and enjoy party all the time, and read and do some arts or whatever you like. Then there is no selection. When there is no perturbation, you don't migrate. You feel, hey, here is the West Virginia is heaven. That's what you put to your car sticker and happily get around. That means you are living happily here. And then if there is no perturbation, there are no mutations. The new mutations will form. If those mutations are beneficial, you keep those mutations. If the mutations are harmful, those babies will die. If the mutations are kind of beneficial, that will spread very fast, because inadvertently you will meet people having that kind of features. Those features are coming from a mutation, for example. And then not inbreeding is happening. If the population is assortative, same kind of people are mating, then what can happen? The more heterozygosity will dissolve and recessive genes will expose, recessive alleles will expose. The populations are small. Say there are only 100 families. They make babies and they mate, and further next generation, they make the babies. Eventually, what will happen? Because there is no migration, nobody is coming from outside, mutation rate is low, then all the recessive alleles, deleterious recessive alleles will expose, and they become inbreds, like inbreds, like having brother and sister mating. And then that is very harmful. So the population has to be large. Okay. whether it is insect population, bird population, plant population, if the ideal population size should be large, and then the mating should happen randomly. It's not that way you can prevent inbreeding and you can maintain heterozygosity. And then there should not be any mutations. Mutations can perturb equilibrium. Like if more genotypes are mutating towards recessive types for some reason, and then our recessive types are increasing because of some selection or because some benefit there, the dominant people are dying, just like in the HIV. And then what happens? It's not ideal population. You will have only one kind of people. Many diverse people will get eliminated. And the migration, if the migration is there in the ideal, if the population is already ideal, there need not be migration. And what happens if the migration happens? The new alleles will enter into the gene pool. They will bring the diverse people or diverse individuals or diverse insect populations. Whatever species we are discussing, the new populations will enter into into a population, into a place, into a geographical region. There, the migration will create more alleles, more diverse alleles. Okay. Now, diversity will increase in a population. So that perturbs the ideal population. Your population is already ideal. You have large population, random mating is happening, no selection is occurring, so no problems are happening. You are all living hale and healthy, beautiful place. And then there is no migration is not needed. If the migration is there, then that is going to perturb the population. That population is going to eventually genetically change. Okay. So this is population genetics. And then you can understand the implications. When you, for any particular locus, you can do the PCR in a large population and understand homozygotes of both kinds, recessive and dominant kind, heterozygotes. If it is equilibrium, heterozygotes will be exactly half. Genotype frequency of heterozygotes is green here. That is exactly half. And then recessive population is Q square, and then dominant population, that is capital A is P square. This is this typical scenario of what? Equilibrium. So what if this heterozygotes are reducing? That means what? That more inbreeding is happening. If more inbreeding is happening, then recessive alleles that are hidden are exposed. If that recessive allele is of a particular deleterious phenotype, and you will see the deleterious individuals in the phenotype. You see, in olden days, in Kentucky and West Virginia, the populations were subpopulations very, very small. Whatever, I don't know much of the history, but you type on the YouTube, blue people of Kentucky are inbreeding in West Virginia. In olden days, you can see the effects of inbreeding. Even that inbreds of Kentucky are still even now alive. You can see some YouTube videos. They are mentally retarded and all. So what has happened there? The populations are small. Lot of inbreeding happened. Because of inbreeding, the genes, the recessive alleles, deleterious alleles that have to be in heterozygous condition are exposed. And then once the people with a lot of recessive alleles, exposed alleles are there, they are all kind of very unique. They are just like inbreds. Their intellectual ability is less. They are susceptible to various diseases. Their upkeeping is very bad. You can see how their lifestyle is. So these forces will operate so fast. So the diverse populations are good, provided the equilibrium is maintained. Okay. Yeah. So multiple alleles are there, like blood groups. So even multiple alleles frequencies also, some people are A group, some people are B group blood, some people are AB group blood, O group blood. Even in case of multiple alleles, you can estimate what is the, now the P is different kinds, A group blood, B group blood, O group blood, AB group blood. So now PI to PJ are different kinds of alleles. Now you can estimate the frequencies of each particular A group blood frequency, B group blood frequency, O group blood frequency, AB group blood frequency. Like that you can estimate the frequencies, and then allele frequencies also can be estimated from that. So even, and if it is X-linked genes, if it is female, they will have just like normally 2H, because they have two X chromosomes, females. If it is animals or humans, we have exactly two copies of the X chromosome. So they follow the same individuals. But if it is males, it's only one X chromosome. So in males, for example, if the Q recessive allele is of some deleterious effect, like color blindness or albino, or any trait like that, a recessive trait, that is going to spread fast in the males comparatively to females, or hemophilia. So in males, if there is any disease, that is not going to be hidden in the heterozygous condition like in females, that get exposed so fast. Okay. So we almost discussed 7:30, holy cow, and no questions are there. Are there any questions at this point? Let us discuss. This is crazy topic. R.D. Weinberg equilibrium is so true. Yeah. If there is any questions, we will start talking about it. Or any dumb question, any simple question can be asked. Yeah, there is nothing like a simple question. A very funny question can be asked. This is so true, how the biodiversity is maintained in various species and why it is important. If there is no biodiversity, then there is very narrow genetic diversity. Narrow genetic diversity leads lesser heterozygotes, and lesser heterozygotes will expose more recessive alleles, homozygous recessive alleles. Recessive alleles generally linked to various deleterious features. That's why the people can become very sick. Okay. So that's why studying the population genetics is very important. You can understand studying what is population genetics. It makes you to understand evolution, and then phylogeny, evolutionary relationships, and then various importance of various loci, kinds of mutations. So it is a very important field of genetics. So what is the difference between Mendelian genetics and population genetics? There you are talking about only two individuals, round seed, wrinkled seed, or any just features like that. But here you are talking at the level of population. It has more applications. If you want to do plant breeding or animal breeding, you need to know at the level of population how the population behaves. Then you can select better horse, better chicken, better cows, right? So if I am giving you just one chick and say this is all you have, and then what kind of chicken you will have? Very dirty chicken. Just same chicken, you breed it, and then it's no genetic diversity. It gets all kinds of diseases, and you have to consume inferior chicken or beef. But you have diverse parents, a population, then plant breeders can make crosses and identify at the level of population what they are selecting, understanding the genetic gain. and maintain the diversity in the population. And they can keep up, the populations in balance and perform the breeding, and can develop multiple varieties or multiple types of cattle to keep up the needs of the people. So one important feature is DNA typing. This we will understand next class. If you have any questions, you can ask. So rest of the topic I will discuss next class. Yeah, almost half of the chapter. Yes, that's good. We did good today. Natasha is asking, would genetic drift directly impact population genetics? Absolutely. If the genetic drift causes populations to break up, then their deleterious alleles get exposed. And then if there is any pathogenicity or something just like a COVID, you see in class, we voted that day how many ever got COVID and how many always got COVID. So if there is no genetic, there is genetic drift, there is narrow genetic diversity, then the entire population get eliminated before the therapeutics is identified. So that directly impacts population genetics. Population genetics is a study of populations and genes in population. It's a very good question. Vijay is asking about the past chapter. Do plants suffering from allopolyploidy keep all the chromosomes from their ancestral species in their cells, or do they lose them over time? If they lose them, what percentage of their ancestors can they keep? This is brilliant question, Vijay. Once two genomes are coming together, like two species are mating accidentally and producing the baby, and that baby is surviving, because the chromosome number is doubled or endo-reduplication happens, whatever way a new species is raised, that has lot of challenges. So some of the chromosomes, then the population needs to increase, and then that population needs to get subject itself to natural selection. So if there is any beneficial effects comparatively to normal species, diploid species, this polyploidy will get that advantage of added number of chromosomes. So sometimes some of the genes will get eliminated and some of the genes will be gained. Actually, you imagine two different species-specific chromosomes are coming into single nucleus. That's not a joke. Then the chromosomes will undergo lot of unequal crossovers because species A, species B, they are still similar to some extent because they had common ancestor. Now they became species and reproductively different. And again, this mating brought them back together, making it as a polyploid. So chromosomes specific to A, chromosomes specific to B, undergo lot of unequal recombination, purging out the segments, creating a lot of diversity within that population, purging the segments and then modifying the chromosomes, multiplying the domains, or losing some domains of the genes, clusters of the genes. So lot of process will happen. That's why the wheat that is probably 5,000 years back wheat is different from current wheat. The paleogenetics is people identifying wheat grains from the pyramids or different places, and then sequencing that older seeds and comparing with the new seeds and seeing that what might how the people kind of bred upon, or the species how they changed from within 5,000 to 10,000 years. You can see drastic differences. I have a lot of pictures like watermelons of 11th century and now. And also humans, say 100 years back or 200 years back, humans are quite different from now. Their physical potential is different. Their response to environment is different. So it's all the time humans are different. We undergo a lot of epigenetics. That is, we store a lot of stress in our genomes. That is another aspect. So if any stress is happening to you, you will transmit that stress to your progeny. So say thousands of years back, the people had all kinds of exposure to the environment, exposure to life challenges. That gets embedded into the modern genomes eventually. So 500 years back, humans are quite different from what we have now. Probably the way we understand things, the way we do things, the way we respond, the way we eat food, the way we digest things, the way we metabolize everything. Altogether, we are different now. It's the life is such a dynamic process. And from now to probably 1,000 years from now, it's absolutely even different. So studying the population genetics and tracking this information and how we store, how we function at molecular level is so important to understand human life or any life that is necessary for diversity. That's a really great question, very intriguing question, Liji. So if you are done with the class is adjourned. We will meet on Monday. You all have a great evening. నా క్రెడిట్ కార్డు వాడి దగ్గర ఉంది, కార్లోస్ దగ్గర. క్రెడిట్ కార్డ్. Carlos has to give me back. Chila.